form their bud, how they then are transported to their destination membrane, and then how they actually fuse. So that's what we're going to get into the details of now. And write, again, this question, what's cytosolic, always cytosolic, what's non-cytosolic, always non-cytosolic. So that's kind of referring specifically to these sides of the protein that we see embedded into this membrane, right? Once this side of this membrane protein is non-cytosolic, it is going to stay non-cytosolic, right? And so the reasons for that are proteins don't switch around. Once they're embedded into a membrane, they don't turn, right? They don't turn, they don't switch their orientation. And then when vesicles fuse, they maintain that orientation because the outer membrane is always going to fuse with the outer membrane, inner membrane always fuses with the inner membrane. So it maintains that orientation. So if I ask you about why is the oligosaccharide on that transmembrane, why is that only non-cytosolic? What would the first part of the answer be?
Addition happens in the lumen.
Addition happens in the lumen, right? So addition happens in the lumen of the ER, that's non-cytosolic. And then we know that protein's not going to switch orientations, so its orientation is going to be maintained because it doesn't move around. And then when it is trafficked in a vesicle, the way that the membranes fuse maintains that orientation. Does that all make sense? Okay, it's very foundational stuff. So let's first talk about budding and these sweet proteins called coproteins. So in one of the videos, it showed a vesicle budding. And that's gonna help you visualize this much more than a 2D, right, static image, or what I can do by making weird claw-shaped hands. But what happens with these coproteins is they have to be recruited to the membrane where a vesicle is forming, right? So part of that is, you notice at the beginning of this image, you have a receptor. This is a cargo receptor, right? It's going to bind to a protein that needs to get trafficked in a vesicle. Once that binding occurs, it initiates this cascade of events that leads to the recruitment of the coprotein. So the coproteins, when they're recruited, the conformation that they want to form is basically this spherical, like lattice-like shape. Right? I think I said this last time. Looking at these always reminds me of like those jungle gym structures, those dome structures at playgrounds that you climb on. I know playgrounds well these days. You guys might not remember. I hang out at them a lot. So when these coproteins form, they get recruited, they start to form this lattice-like structure, and it just pulls that membrane away into a bud. Right? And so what happens is it ends up forming a sphere, right? Now it's created its vesicle, it's ripped that membrane away and formed this little membranous sphere, right? I.e. a vesicle. And then once the coproteins have done their job, they're triggered to disassemble. So they're done. The only thing the coproteins do is help in that initial formation step, the budding of that vesicle from the membrane. Yeah.
So when one coprotein binds, does it increase the affinity for others to bind as well? Or?
No, it's really, well, the coproteins are really recruited by whatever kind of cargo receptors or receptors on the surface of the protein. And once you get kind of a critical mass of them, they start to form this specific structure that pulls the membrane away.
Okay.
And there, in the video, if you watch the video, it's a lot more complex than just coproteins. There's a bunch of other accessory proteins that help the coproteins do their job. And there are, so far, 3 identified coproteins. There could be more that we just haven't identified yet, but right now, we know of 3. And here are the things that we know about the coproteins. So first there's COP1 and COP2, and then there's also COP2. We're going to talk about where we typically see those coproteins forming vesicles. So COP2 is always involved in forming vesicles on the ER membrane. Vesicles form on the ER membrane, you know for sure. The coprotein involved is COP2. So a COMP2 coated vesicle, it's next, you know it came from the ER membrane, and you know its next stop is going to be the Golgi. Because that's where vesicles go from the ER. Golgi. All right. Now from the Golgi, there are two potential coproteins involved. COP1 and clathrin. And there's a lot of messiness with arrows happening on here. And that's just kind of to show that vesicles go in a lot of different directions to a lot of different places in the cell. Right, so far we've heard COP2 from the ER. COP1 are vesicles that are budding from the Golgi. Clathrin vesicles can also bud from the Golgi. And you also see during endocytosis, the vesicles that form from the plasma membrane and move inward, those involve clathrin. Yes?
Are vesicles just kind of like tools used by the site? They're different functions.
For trafficking, yeah. Vesicles are little tiny, right? They're little tiny vesicular Ubers that just traffic things around, whether it's lipids or proteins or other molecules. Yep. So they're just associated with trafficking. Okay. Or lift. Uber, I don't know if you know about that organization. There's some help. Little turkey in there. All right, so again, COP2 from ER, COP1 from Golgi. Clathrin could be from Golgi or from plasma membrane. All right, so yeah. Oh no, so COP1, they see COP1 sometimes in outward flow too. Yeah.
COP2, there's COP1, and if it's just if you're on site, then it's taken out.
No, that's a little bit more detail than I want you guys to think about with coproteins, because the COP1 coated vesicles can also go back to the ER. So they're not necessarily, the only coproteins that's definitively associated with a direction, COP2 is always going to be an outward flow because it's moving from the ER out. And then clathrin at the plasma membrane is involved in inward flow. But then COP1 and clathrin can do all sorts of crazy things from the ER, right? From the Golgi. From the Golgi. You're recording. Rewind, erase.
If you do like a U-turn, you know about COP2.
Yeah. COP1. You're doing it now too. Yeah, COP1 leaving from the Golgi, and it might go back to the ER or it might go outward. So these are the most common functions associated with these coproteins. The variation can get a little bit crazy and hard to fit into a nice contained box, right? So it seems like there's a lot, there is still a lot for us to understand about the variation in coproteins and, but this is all I want you to understand now. It's kind of the basics of where we know we see these coproteins aiding in the formation of vesicles. So they're different they're different proteins, right? They have different names, but they're performing that same function to pull membrane away from like a source membrane and turn it into a tiny little vesicle. Okay. So another sweet protein that I want to talk about today is dynamin. So dynamin is always associated with clathrin-coated vesicles. Okay. And so what happens with clathrin is it pulls the vesicles into a bud, but if it doesn't have dynamin there to like bring the neck of the bud just stays associated with the membrane. It can't pinch off unless dynamin grabs onto the neck of the bud and helps pinch it off. And so scientists discovered dynamin because So Diamond assembles into a ring around the neck of the forming bud. It recruits the help of other proteins, right, the and associated proteins, and basically just ring the neck of that forming bud and cause it to release from that membrane. So yeah, go ahead.
So if there is no diamond, the buds would not be able to bud off.
You got it, the buds would form, but you wouldn't see them leaving the membrane. And so that's what scientists saw in this case, right? So here's a membrane that has a Dynamin mutation, and that's preventing the function of Dynamin, right? So you see all these buds forming, so the buds can form, but you don't see any vesicles out here, right? So they're not able to release from the membrane. So what would happen because then scientists were like, okay, let's see what we can do if we increase the activity of Dynamin. What would you expect to see? See like way more vesicles than usual, be like souped up vesicle formation. So that's what scientists do to determine the function of a protein, right? They create mutations where they block the activity of a protein and then go, okay, what happens? And the phenotype that results, like, okay, that's what this protein was doing, right? It was helping in this cellular process that we're no longer seeing happen. Or they create gain-of-function mutations where it soups up the activity of the protein. Yeah.
Is the diamond found in the cytosol or is it found on the actual plasma membrane?
That is a good question. So is it always associated with the membrane or is it specifically recruited there? I'll look that up. I know for sure the accessory proteins have to get recruited from the cytosol. I think the Dynamin undergoes the same recruitment process as its co-proteins, but I'll double check that. Yeah.
So for testing purposes, if you had like asked if there was no Dynamin, could you say that there would be like a lack of like lipids being carried out or something because of or a lack of vesicles?
Yeah, and so specifically what vesicles would you see affected? You would see vesicles at the plasma membrane affected, and then some of the vesicles that form at the Golgi affected, because that's where we know clathrin is important. So that's actually a good segue to, and if I didn't completely answer your question, actually tell me now, did I completely answer your question? Kind of, but please no. So if you were to block the activity of clathrin, what would you see? Do you see vesicles at all? I'm gonna take that eyebrow raise as a no. Correct. No. If you blocked clathrin's ability to function, you would not see vesicles forming at the plasma membrane, right? Because clathrin is what gets these buds to form. So that's different than what we're seeing now, right? Clathrin is important for forming this structure. The Dynamin is important for pinching that vesicle off, causing it to release from the membrane. So you would see an actual vesicle right here. The vesicles here. Does that make sense? Okay. So, yeah.
Does it only prevent the formation of vesicles coming into the cell or also out?
It would affect, so at the plasma membrane it's only coming in because vesicles, when they fuse with the plasma membrane, so vesicle formation is very different than vesicle fusion with the plasma membrane, right? So formation, this is the beginning formation of a vesicle. So, right, so it only affects inward flow from the plasma membrane, but for vesicles that fuse with the plasma membrane, that's dependent on snares. Yeah.
What did Pop 1 and Pop 2 have something like that, or do they not leave?
It seems like they don't, but that doesn't, it could just mean, I mean, maybe in some cells they do, right? We're talking about eukaryotic cell biology, so we're generalizing what happens in every eukaryotic cell across every eukaryotic organism. Right. So maybe in some organisms they do, they just haven't been identified. And so initially, people wondered, okay, what's up with Dynamin? Why does clathrin need Dynamin? And do you guys remember what's different about the features of the plasma membrane than other membranes? It has more of something. I think I heard someone just say it. Maybe not. Yeah, cholesterol, which makes it more what? Rigid, yeah. So they were thinking that clathrin, right, it's doing its best to pull out that membrane, but because of the rigid nature conferred by cholesterol, it couldn't pull it out all by itself, and so it required this Dynamin to pinch it off. But then they noticed Dynamin was also helping it at the Golgi, which is not nearly as rigid. And so then they said, okay, well maybe just clathrin, the way it developed was it always needs Dynamin to help these vesicles, but so it could just be that the clathrin itself, the kind of intermolecular forces between the clathrin components that form this lattice just aren't strong enough to pull the bud apart, whether it's rigid membrane or not. Yeah.
So for comp 1 and comp 2, how do they bud off without Dynamin? Do they have like
They're super strong, man. They pump their co-protein weights. They don't, yeah, they just, for whatever reason, they can pull off that vesicle all on their own. Is what it seems like. Yeah, it could be that there's some protein that's doing the same thing as Dynamin, we just haven't identified it yet. Alright. Alright. I'm insecure now. Never apologize for asking questions.
Comp 1 and Comp 2, they don't have that pinching aspect, okay.
They're just able to pull off the bud all on their own. Yep. As far as we know, right? Always remember that in science. What I'm teaching you is as far as we know. This is what we know. That doesn't mean it's not going to be upended and turned on its head tomorrow by some paper that gets published by some lab that's figured out a similar process or problem. But right, that's what we're working with. I'm teaching you foundation. You just know that exceptions will arise. That makes it fun and exciting, not confusing. Okay, so now we're going to go on to transport and targeting, right? So we have our vesicle formed, the co-proteins are gone, the vesicles don't just go wee and swim to their destination membrane, it's very controlled. So how does that happen? All right, so remember, on the surface of the vesicle, it's facing the cytosol, so it has that cytosolic submembrane meshwork outside of that vesicle. So let's draw vesicle with stuff in it. Let's get technical. Maybe there are receptors themselves that are getting transported. So TM stands for transmembrane. Sorry, that got really fast. I'm sure you were laughing.
What is the X's around it?
What's that?
The X's?
Oh, submembrane meshwork.
Oh, okay.
Well, all of it is a submembrane meshwork. I just always draw actin as actin filaments as this meshwork. And this will make a lot more sense once we go over our cytoskeleton lecture. Skeleton. Okay, so basically everything that's on the cytosolic side of this vesicle is considered the submembrane meshwork. And that's what is seen by anything in the cytosol. Anything inside the vesicle is neatly contained by that membrane. Right? It's neatly contained by the membrane. So if you're talking about transporting this vesicle, the things that attach to it are going to be attaching via the submembrane meshwork, is the take home. Okay. So, protein signatures. This is a vesicle protein signature. It's just referring to everything in the submembrane network. It is what the protein signature of a vesicle is. It's a super general term that would help, like a protein signature, right? It would differentiate one vesicle from another, right? Because it has its own protein signature, its own thing. Because any individual vesicle might be transporting something different. If you had two vesicles that had the exact same cargo and budded from the exact same membrane, they would probably have the exact same protein signature on the cytosolic side of that vesicle. Devin.
Same protein being what?
Transmembrane protein.
Being, it's like,
It's-
stop like sentence.
What's that?
You stop like mid sentence. Being what? Just being.
I was gonna, just being, just existing. Molecular biology, philosophy, being transported. Thank you, sir.
I was just like, is it just having a good time?
God, you guys want me to finish sentences?
Yes.
Okay, so here's the cytosol, right? So we want this vesicle to get transported somewhere. So there are the transmembrane proteins, right, would be cytosolic side of transmembrane protein. So this is a much better looking transmembrane protein than my green piece of crap over there. But right, so the cytosolic side of this protein is exposed. So it can be seen by cytosolic factors, right? That's all you need to appreciate now. And then also any peripheral proteins, right? So there are going to be proteins that are like the actin cytoskeleton is peripherally associated, and there's going to be other proteins that might have lipid anchors that are peripherally associated. Membranes are messy. There's lots of stuff on them. Right, so you have the lipid bilayer, you have any proteins embedded into that lipid bilayer, and then any other proteins, peripherally associated with the membrane. With me? There's take home, and that's any membrane. You have lots of stuff associated with it. And a vesicle is just a small piece of membrane, so it's also going to have a lot of stuff associated with it. So any vesicle that buds from its source membrane, what's on the outside of it is what helps connect it to motor proteins, help transport it. So peripheral proteins make up the submembrane meshwork, and then signal patches. So remember when I introduced the idea of signal peptide versus signal patch? A signal peptide, that's like we're already done with the part where signal peptides are important for protein trafficking. That happens right after translation, right? ER signal peptide, immediately recognized by SRP, helps translocate that whole translation initiation complex to the membrane. Or, right, mitochondrial, the mitochondrial pre-sequence, that's its signal peptide that gets those proteins trafficked to the mitochondria. Peptides are continuous stretches of amino acids to help indicate right at the time of translation where that protein needs to go. So signal patches are different because they can be kind of located far and wide. If you have the primary sequence of that peptide stretched out, and then they're brought together after tertiary folding. So if this protein had has a signal patch, it's these two sequences that form a specific conformation once the protein folds, right? And so what they found with vesicles is that there are specific signal patches on proteins that can help associate with motor binding proteins. Motor binding proteins are proteins that bind to motor proteins. I did not name these things, not kill the messenger, right? So what they found is, so this is an example of a signal patch. as part of the cytosolic phase of the vesicle, you can have signal patches or cargo receptors or other peripheral membrane proteins that link to motor binding proteins. And again, motor binding proteins are what actually right? So motor proteins are this cool family of proteins that walk along the cytoskeletal filaments. That's how transport happens in cells. So anytime you hear, bless you, any type of transport that's happening, people might be leaving out the motor proteins and the cytoskeletal filaments, but its odds are pretty stinking good that a motor protein is attached to whatever it is being transported and it's walking along the cytoskeletal filament. Yeah?
So for the non-bound free ribosomes, do they also have signal pouches that attach to motor proteins, or are those only transported by the strand?
Usually it is the signal peptides themselves that associate with the transport protein,
Okay.
and that transport protein.
Okay, so they don't use like the motor protein highways.
They do. They just write like if it's a mitochondrial protein. Well, there's two lines of thinking, and it's not. I'll look it up to see if there's clarity in the field now. But basically, the transport protein would bind to the mitochondrial pre-sequence, and one of two things happens. Either the mitochondria are so densely populated in the cell that transport protein, it doesn't really have to go very far. Odds are better that that transport protein is connecting to a motor protein and helping take it there.
Okay.
Yeah. Just gonna draw a motor protein really quick. Motor protein, motor domains, light speed. Cargo binding. And then it uses cytoskeleton as tracks. Why? And so the motor proteins will walk in specific directions alongside a skeletal component. So this is the part, right, that would bind to motor binding protein, and that's the part that's bound to vesicle. Yeah, no.
How does it decide the direction to go in? And what would happen if it met another motor protein going in the opposite? Or is it all one direction and then another track is?
Yep. All motor proteins are designed to go in one direction, and also remember it's 3 dimensions, so like you get another motor protein right here underneath. Good. I'll leave that up there too. Sneak a peek.
So that motor binding protein that's not signal pouch, or that?
It's what associates with the signal pouch. Okay. All right, so the vesicle protein signature, right? Something on the outside of that vesicle is going to include the motor binding proteins, and that's what binds to the actual motor proteins themselves. The 3 different types of motor proteins are kinesin, dynein, and myosin, and we'll learn more about those in the cytoskeletal lecture. You'll get to learn about which cytoskeletal filaments they bind to and what direction they move on. It's going to be exciting. Okay, so right, here's the motor protein, and yeah, so one phenomenon that happens a lot when vesicles are super similar, once they decoat, you'll see them fuse, right? Because all their membranes are really similar to one another, and you'll see this fusion event with one of these, what are called vesicular tubular clusters. Don't worry, you will not see the term vesicular tubular cluster on my exam. I'm going to ask about it in the review questions. But if you want to talk about them in office hours, think about this hours. And so it's connected to this motor protein via motor binding protein, right? So the motor binding protein would be right here at the cargo binding tail. You can see in this case it's walking along a microtubule. And so if you're going from the ER, if you're leaving the ER and you're moving to the Golgi, you're first going to hit the cis-Golgi network. So the Golgi is a series of these stacks that are called cisternae, and it goes from cis to trans. The cis space is where vesicles are received, and we're going to talk more about that in a few slides, but just your primer to that. The cis-Golgi network is where those top two coded vesicles, once they decode and then get trafficked to the Golgi, they fuse with the cis space of the Golgi. So CIS, cis space. Okay, so that's transporting, so it moves it, but still, that vesicle might bump, if it bumps into the wrong membrane, it's not going to necessarily fuse with it because it won't get docked there. So the way it gets docked and then actually fuses, you need RABs for targeting and you need snares for fusion. Okay, so the cytosolic protein signature is also going to include these proteins, the RABs and the snares. So we're going to add to our protein signatures. Not mess these up. You're also going to have so, targeting and tethering. It's a function associated with RABs. So RABs, basically, here we have our RAB, and it has a target proteins on any potential target membrane called RAB effector proteins. And so target membranes, whether it's in the ER, the Golgi, the lysosome, the plasma membranes, they all have their own RAB effector protein. Right? And so I think in a few slides there's a chart where it shows like RAB2, RAB3, RAB4, RAB5, and all of the different RAB proteins that are specific for these membranes. So it's a specific partnership. There are a bunch of different RABs associated with vesicles, and those get recruited to the vesicle when they form. And then they pair up with specific RABs on the target membrane. And you have to have that precise coupling in order to get a vesicle to basically get docked, right? To be tethered to that membrane. So that's what gets it there. But the RAB association alone isn't sufficient to cause membrane fusion. So RABs are for membrane targeting. So if you had a vesicle and you wanted to mess up the target membrane that it reached, you just change the RABs. Change the RABs and you're going to totally change what place in the cell that vesicle gets docked to. Okay. And then for actual fusion occurring, it's these red and blue proteins that have a passionate relationship. They bind each other so tightly that they essentially squeeze out all of the water molecules. And so if you squeeze out the water molecules, those lipids aren't afraid to change their conformation, right? Because for lipid fusion to occur, those lipids that are fusing need to kind of do little lipid gymnastics. And if you still have water molecules around, right, those fatty acid tails are like, I don't think so. So what the snares do is they bring, and I think I have an image coming up, they bring those membranes so close to one another that it dislocates water molecules and allows those lipids to kind of move freely and commingle with one another. So snares really cause the fusion event. You need all these components, but they're important for very specific pieces of vesicular trafficking. So you need budding to occur properly, that's coproteins. You need transport to occur, right? That's the motor binding proteins, the motor proteins. You need to get targeted properly, that's RAS, and you need it to actually, the membranes to fuse, that's snares. Right, so here's the RAS, and here's the list of the membranes that they're associated with. Please do not memorize these, right? You can Google that if you really want to know what RAB is associated with what target membrane. The take home is, right, different RABs target vesicles to different cellular locations. Right? And so, of course, the cell has a bunch of different RABs. There's a lot of different potential locations to transport those vesicles. These are the address, right? It's the address getting slapped on the outside of that vesicle. Okay, good. I was afraid I took this slide out. So snares catalyze the fusion of membranes, right? So what happens, the snares, when they interact with one another, they pull the membrane so close, right? It dislocates the water molecules, and that's really what allows these lipids, when they interact with one another, to change conformation, so that they can form one continuous membrane. And so, right, there has to be a good match between lipids, and this kind of gets back to the whole lipid signature of the outer and inner leaflet. Whenever you see membranes drawn in a cell biology or cell molecular biology course, and they're talking about what's happening in the membranes, you're always going to see a difference in the outer and inner layer of those lipids, because that's an accurate depiction of those membranes, and that have different lipid compositions. So because of those lipid compositions, it directs the way fusion occurs. You always have the cytosolic leaflet fusing with the cytosolic leaflet, and non-cytosolic fusing with non-cytosolic. Always. So the lipid signatures have to match in order for fusion to occur. Right? Okay. So lipid signatures, okay, right. So someone was asking me about the question that related to this on the review question, and I thought the wording could have been a little bit more clear. It was asking about the composition of lipids and proteins, and this is the slide that I was wanting you to think about. It's just that the non-cytosolic and cytosolic symmetry, well, it's asymmetric, right? On either side of the membrane, if you're thinking about lipids or thinking about proteins. So for lipids, hopefully I've driven this point home enough by now that you appreciate that there are a whole bunch of different types of lipids, and the lipids that exist within the non-cytosolic layer of a lipid bilayer are going to be distinct from the lipids that are in facing the cytosol, right? There's a difference. It's not symmetric. Asymmetric. You see different lipid compositions. Right? That's really how, like, hopefully when I ask why, the why part of it is it's really important for directing vesicular trafficking. It's really important for a bunch of other functions that we haven't talked about so much yet, but I'll get at it when we talk about lysosomal trafficking. I'll touch on that. So in membranes, right, the lipid signature is asymmetric. Protein signature is also asymmetric. So if you were looking at a protein that was embedded in this membrane, you would be able to immediately tell me which was cytosolic and which was non-cytosolic, Because what is different about the cytosolic side of transmembrane proteins? What's there that wouldn't be on the cytosolic side? Oligosaccharide, right? So those sugar groups, they get added in the ER lumen to the non-cytosolic side of a protein. So proteins that are trafficked to the plasma membrane, okay, when you see that ER lumen, get that sugar added to the non-cytosolic side. Once it gets out to any membrane, you're always going to see that sugar group on the non-cytosolic side of the protein. So that's what I mean there. So this is partly because oligosaccharides, the way that they're added is totally asymmetrical. All right, and then here's another phenomenon that's really interesting. Proteins that live within a membrane. So you notice that, right, even though it's that you may have distinct phospholipids on either side of the membrane, there are a bunch of different phospholipids there. Well, there are transmembrane proteins that might attract or repel specific phospholipids. So you'll start to see certain proteins creating what are called lipid rafts. So you'll create an area of lipids that is different than the rest of the membrane, right? So say I'm a transmembrane protein and I attract sphingomyelin but repel phosphatidylcholine. All of a sudden you'd see a domain where there's a bunch of sphingomyelin and not as much phosphatidylcholine, right? So proteins, all molecules are going to attract or repel or have maybe a neutral interaction with other molecules, right? So proteins can absolutely attract or repel phospholipids in the membrane. They can totally influence what phospholipids sit around them in the membrane. Does that make sense? Okay. All right. Who wants to talk Golgi? Who doesn't want to talk Golgi, am I right? Oh, sorry. It's a really fun job. It's a really hard job, but it's really fun because in most social situations, when I'm silly like this, my friends are like, but I'm a captive audience. I can be as silly as I want, and no one in here is going to be like, you're being lame. On my evaluations, you might though. I might sit there. Okay, so maybe I'll turn it down. All right, the Golgi apparatus. The Golgi apparatus is always described with some food analogy, right? It's either like a stack of pita or a stack of pancakes, because it's composed of these cisternae, right? These kind of membrane enclosed long structures, all stacked on top of one another. And so, right, I mentioned before that when vesicles leave the ER, they're immediately going to go to the Golgi where they undergo more covalent modifications. So in the ER, yeah, they get their initial in-link glycosylation, but that Fibble sugar is going to get modified a ton in the Golgi. There's a bunch of other modifications that can occur there. So right, I always liken it to a protein getting its finishing touches, like the rest of its clothes on, or getting its hair done, if you will. That has to occur before the protein is ready to take on its final conformation, so it can go have whatever function that has in the cell. Right, so in the Golgi, you get covalent modifications, including all this great glycosylation, and then sorting occurs when proteins leave the Golgi, right? Because it has to sort those vesicles. The vesicles have to be packaged up with the right Rabs to get into the right destination. Are they going to go to the plasma membrane? Are they going to go to a lysosome? Do they need to go to an endosome first? So those that packaging occurs in what's called the trans phase of the Golgi. So it goes cis to trans. So again, cis phase, right, is going to be closest to the ER. Vesicles leave the ER, fuse with the cis phase. Proteins move through the stack of cisternae, and then they get they bud in a new vesicle and leave the trans phase to go to their final destination. Okay, so far. So TGN, right, that It's interesting. So you see the trans-Golgi network abbreviated all the time as TGN. The cis space, its abbreviation would be CGN. I usually hear people call it the cis space and then the TGN, which is not consistent, but that's just the most common lingo you hear. Okay, cis to trans. So oligosaccharide processing, right? It's one of the most important processing steps that occurs, and it's in this really prescribed manner, right? And you don't have to remember each of these steps that occur. I just want you to appreciate that what happens in each cisternae is predictable, right? So it's, I think that's the term I'm looking for. It's, do I have it here? It's like an assembly line. I don't have it here, but it's like an assembly line. So a multi-stage processing unit, that's a much fancier way of saying assembly line, right? So as the protein moves through, there's this sequence of events that occurs, these sequence of different modifications that occur in successive stages as it moves through the pancake stack. It moves through the Golgi. And then, right, the final thing that happens is things get sorted. And we're going to talk specifically how lysosomal proteins get sorted to ensure appropriate delivery to the lysosome. Yeah.
So in chapter 10, when we were talking about combinatorial regulatory code, is that what's occurring here? Like all the phosphorylation and all those are going on?
That is a really great question.
Separate?
Not exactly. So I was kind of talking about proteins. This is in the beginning, proteins just getting produced in the first place. And then those covalent, these are covalent modifications, so they fall under the same category of covalent modifications.
Yeah.
That was specifically about you have a protein that's done being made, it's at its home where it's going to do its job.
Okay.
You can phosphorylate it by a cytosolic kinase and or dephosphorylate it or acetylate it or deacetylate it. So this is just the beginning, the protein getting dressed. That's talking about like the protein's already where it needs to go and someone puts a hat on it, right? Or puts a jacket on, right? It can still be modified after it's done being processed.
You like more preliminary?
Yes,
processing?
exactly. These are preliminary processing. It's just being born.
Okay.
The protein is just being born, and then once it is it's born and it's at its location where it's going to function, the way you can function a fully mature proteins, the way you can manipulate or change a mature protein's function is different covalent modifications. Right? So like P53, the example I use, that's actually a cytosolic protein. So that protein is translated on a free ribosome. It never even sees the Golgi. Right? So cytosolic proteins that are modified on free ribosomes, they don't get trafficked to the ER, they don't get trafficked through the Golgi, right? They just get translated on a free ribosome and then they're in the cytosol where they're going to do their job. And so P53, right, it has all these different jobs, and the way it's directed to do one job or the other is through covalent modifications as a mature protein.
Okay.
This is important, so I don't want to go on until you're like, yes, I get it, because that seemed like
So like,
just stop talking to me.
what are these things? Like, what's going on here? Like, in terms of like how preliminary it is.
Yeah. So the protein has just been translated, right, into the ER. It's done its initial folding and had its initial oligosaccharide added to the asparagine residues, right, because we're talking N-links. And then once it moves to the Golgi, one of the big deals that happens is that oligosaccharide gets processed in a bunch of different ways. And in fact, the other modifications so most of these, so you can, there's a specific step that happens to lysosomal proteins that we're going to talk about. So it gets this special tag that happens here. And then removal of mannose, that's a change on the oligosaccharide. Removal of mannose, addition of WNAC, that's also a change on the oligosaccharide. The addition of lactose or nana, those are also oligosaccharide specific. So it's really heavily influencing oligosaccharide. Those other modifications, and then sulfation of tyrosines, you can ignore that, we're not going to talk about that. this is really heavy on affecting the oligosaccharide. The other modifications that we talked about, like a specific kinase phosphorylating a protein, that's not happening in the Golgi.
So it's more specific, just like where it's gonna go.
Yeah, it's getting its initial modifications finished, and then sorting it to where it's gonna go. And then when it's where it's going to go, a lot of those proteins have different jobs that they can fulfill, right? So say you are a transmembrane protein that functions in signal transduction. One of the modifications that might happen to you is you get phosphorylated, right? And that triggers your interaction with a downstream protein. So that's something that's just in response to what's happening in the cell at any given time. So we're just talking about proteins being born, right? We talked about how genes get transcribed and then translated, and now how they get delivered to where they're going to live and work. And then as the class proceeds, we're going to talk more about specific proteins doing their job and how how their function can then be influenced while they're doing their job. And maybe a covalent modification is what can help switch a protein from being on or off once it's already been made, or manipulate what protein it interacts with. Does that make sense?
Yeah.
That was more.
So in one of the video clips I watched, it made it seem like the proteins weren't necessarily moving from one to the other, but the cis would slowly move forward to become.
Oh, okay, yeah, okay. Yes. I don't actually talk about this, but that's really cool. There's a lot of active debate in the field. Sarah, right? Crap. There's an A in both, so okay, I'll put it again. Okay. Winning over here. Thanks for being so nice about that. Okay, Christina. So Christina's question, right? This is cool. So for how do vesicles get from here to here, right? And so I'm not going to spend too much time on it because it's not on the test, but it's totally fascinating. So they for a long time they thought a vesicle fused, stuff happened in the cisternae, vesicle forms again, fused with the next cisternae, and so on and so forth. So you keep having a vesicle budding, then or fusing, then forming, then fusing, then forming all the way through. And now what they think is, or the model that's favored is called the cisternal maturation model, where basically you have the vesicular tubular cluster fuses and forms a new cis network. And then that cisternae just keeps moving through, so it matures. And part of how it matures is there's this constant retrograde flow of the enzymes that perform whatever glycosylation event happens. So there's constantly vesicles moving back to create the cis cisternae or medial cisternae. Yeah. There's still a bit of debate in the field about that, but cisternal maturation seems to be the most favored hypothesis. All right, and so when vesicles leave the Golgi, they have new transport, targeting, and fusion information, right? So that's part of the job of the Golgi is to make sure that these vesicles are targeted for the plasma membrane, for the lysosome, and sometimes they end up going to the endosome, which can be intermediate between either the lysosome or the plasma membrane. It's crazy. Yeah. Basically go back to the ER. Usually there's earlier retrograde flow that it goes back to the ER. Okay. So right there, all these different covalent alterations that happen in the Golgi. Most important is the sugar assembly. I really just need to create my own figure because the one in the book is really craptastic. It's kind of confusing. The whole point that they're trying to convey in the image. And then the ER lumen you get the addition of a sugar that kind of is pretty monolithic, like it looks the same on all proteins that get glycosylated in the ER. And then when it moves to the Golgi, all these crazy things happen. So all this change can occur in the Golgi in terms of things, individual sugar monomers being added or taken away. But the big, broad concept that I think is really important to appreciate is that there are hundreds of glycosylating enzymes in humans. And so you can have in different cells, different combinations of glycosylating enzymes in the Golgi. So that you can have the same protein produced, but when it moves through the Golgi in two different cells, the oligosaccharide can have a different structure once it leaves. Okay, so there's variation in those glycosylating enzymes, so you can get cell-specific patterns in the sugar structure that leaves the Golgi. Does that help your brain process it? Yeah, so see, there are two enzymes. Let's call them mannase 1 and mannase 2, or manosidase 1, manosidase 2. So manosidase 1 adds mannoses in a specific way to oligosaccharides, where manosidase 2 adds it in a totally different way to oligosaccharides. So maybe the Golgi in the liver has manosidase 1, and the Golgi in neurons has manosidase 2. So, right? Proteins that are processed in those two different cell types are going to have mannose added differently on the oligosaccharides. Structure can vary. All right. So I'm going to ask you a question about cell specificity. Like Golgis can have cell-specific glycosylating enzymes. You go, yes, that is true. Golgi can have cell-specific glycosylating enzymes. All right. So transport from the trans-Golgi network. So most of the time, people are talking about, right, okay, right, this is what I'm talking about. There are two different ways that vesicles leaving the Golgi can get to the plasma membrane. There are two delivery mechanisms. One is called constitutive. So this means that once the vesicle buds from the Golgi, it's just going straight and fusing with the plasma membrane. It's just automatic, it's going, and this occurs continuously as needed. So if a protein so maybe you're talking about like a skin, a cell in the dermal layer of our skin needs to produce collagen. It's triggered to produce collagen, then collagen gets made and leaves in a vesicle, fuses with the membrane, and just goes down to the extracellular matrix. Right? So as needed, things get made and dumped out there. Regulated secretion is different. It's regulated. So, in highly regulated. So what in this case happens is protein gets produced and forms a vesicle that basically just docks at the membrane. It doesn't fuse. So if that vesicle gets made and it gets targeted to the membrane but doesn't fuse, what protein do you think is waiting for that regulatory signal? Snares. Yes. So my favorite example of this happens in neurons. Of course. So have you ever thought about or learned about what happens when an action potential fires? What happens in the axon? Does anyone know what actually floods into the axon terminal? Calcium. That's right. So action potential on neurons, when it hits the axon terminal, it leads to an influx of calcium. Vesicles that contain neurotransmitters, they're packaged and ready to go, right? Because you're talking synaptic signaling. When that neuron fires, you don't want it to be like, okay, now we need to make dopamine and go through the whole synthetic process, right? What happens is those neurotransmitters are packaged in vesicles and they're just waiting to get their signal diffused. The snares, this is how it works. The snares in those vesicles that neurotransmitters are packaged in, they're dependent on calcium. So they just sit there docked. When the action potential fires, it leads to an influx of calcium. That calcium binds the snare, boom, fusion. And you get a release of those neurotransmitters. That's not the only type of regulated secretion, so there's a lot of examples of regulated secretion, and it's usually reserved for things that you need to be released in a regulated fashion, right? So that it's only determined when the cell gets some secondary signal that says, we need that now. Oh, okay, then it causes fusion and release. Yeah?
Those neurons, is that allosteric sites? Allosteric proteins, or is it some other?
Oh, boom, good, integrated question I don't know the answer to. And I will look up for you. Yeah, boom. Firing lines, yeah.
Is the same thing as for the SR,
For the
sarcoplasmic reticulum?
sarcoplasmic reticulum, oh, in that case, are the calcium and sarcoplasmic reticulum in vesicles? I think they're just free. I think the calcium is just free in the lumen, and it leads to a calcium channel.
Oh.
Yeah. Sorry, I was like, no. Yeah, the calcium in the sarcoplasmic reticulum, that's gated by a calcium channel that opens up. Yeah, I should know that. They're calcium. The calcium channels are ryanodine receptors, which are one of the. So, okay. All right. So this is what it looks like, as determined by the person who makes figures for this textbook. So you have these components that are leaving the Golgi. If it is constitutive, then it's just going to be packaged into a vesicle that's just going to automatically fuse, right, with the membrane that gets delivered out into the cellular matrix. If it's regulated, you get the formation of that secretory vesicle, but it only fuses when some secondary signal binds, right? And that can be, right, the examples are hormone or neurotransmitter. There's a lot of different secondary signals that can trigger the fusion. The point is the fact that this cell, there has to be some other signal that says, okay, we need that secretory vesicle to release its contents now. Good? Lysosome targeting. All right, so I mentioned way back at the start of this lecture that, right, the lysosome is like a garbage disposal of the cell. It has all these enzymes in there that are designed to chew things up. Nucleases, proteases, glycosidases, lipases, right? They're breaking up nucleic acids, proteins, sugars, lipids, everything. These are collectively referred to as acid hydrolases. All these enzymes in the lysosome, they're called acid hydrolases because of the way that they cleave proteins. And also they work specifically at an acidic pH. So acid hydrolases are only active at the low pH of the lysosome. Right? So they're optimally active at a pH of 5. And the cytosol, right, was a pH of apparently 7.2. If these enzymes were released, they might have some activity, but it'd be limited. And it's part of the way someone asked me, how do these lysosomes, when they're first produced, how are they not chewing things up? And that's part of the protective mechanism. They're just not totally active at that pH yet. So these are really deadly to the cell. And so they're made in the ER, right? And then so they're in theory inactive within the ER, and then you want to make sure when you traffic them, you take them right to the lysosome, right? So what's the mechanism the cell has used to make sure these proteins get targeted? It is a tagging system. So all of these lysosomal proteins, which you'll also hear collectively referred to as lysosomal hydrolase, yeah.
Did you say that they're only active at low pH of the cytosol?
If I said that, I did not mean to. I meant of the lysosome. Yeah. So the cytosol is pH 7.2. Lysosome is pH 5. Okay. So there's this tag. The abbreviation is M6P. That stands for mannose-6-phosphate. Really? So here's our lysosomal enzyme, right? Which is a referred to, right, as lysosomal hydrolase, sometimes lysosomal acid hydrolase. And this pitchfork-looking thing is supposed to be its in-linked oligosaccharide, and it gets this very special modification on this 6-carbon ring sugar called a mannose. It gets the addition of a phosphate on this carbon, on the carbon in position 6 of the mannose, right? Hence the mannose 6-phosphate. All right, so it's an addition to an oligosaccharide that is specific to lysosomal hydrolases. That is the marking that denotes this as a lysosomal hydrolase. All right. So they're added to this in-linked oligosaccharide, and that modification happens in the Golgi. N6P is added in the Golgi. And what happens then is in the trans-Golgi network, right, which is where the sorting has to happen, right, the ultimate sorting, there's an N6P receptor. There's a specific receptor that grabs onto the N6P tagged proteins. And once it grabs it, it initiates the formation of a vesicle that will recruit RABs that target it to the lysosome. Okay, and this is a topic that, so the reason I have it on the slide is because if anyone has the book, they're reading about it in the book. It's a little bit messy, though, in terms of what we understand about endosomes. So endosomes are big old sorting stations for the cell, right? And so when things undergo endocytosis into the cell, usually their first stop is an endosome, and then the endosome decides what's going to go where. Is stuff going to get sorted back to the membrane? Is stuff going to the lysosome? And then in outward flow, sometimes when vesicles leave the Golgi, they'll first go to an endosome, and then get sorted to the lysosome. So if you want to learn more about the distinctions between early and late endosome, we can have that conversation in OPSARS. It's just a little bit messy because the field isn't quite sure exactly, is it that early endosomes are just developing into late endosomes and then they fuse with lysosomes, or is it something distinct from that? The part that I really want you to appreciate is the fact that N6P tags get added to lysosomal hydrolases in the Golgi. That N6P tag is bound by a receptor in the trans-Golgi network called the N6P receptor. And that initiates the formation of a vesicle that is going to guarantee delivery to the lysosome. Okay? Okay. All right. So how do these proteins get recognized for addition of the N6P tag? See this part of the protein that's darkened red? Guess what that's representing? Signal patch. That's right. It's a signal patch. So once this is messy, just stay with me. Try. Oh, have some animation. Signal patch. Signal patch on the protein. Once it gets into the Golgi, right? This is happening in the Golgi. There's this enzyme that recognizes the signal patch. That's the enzyme that modifies its sugar, the oligosaccharide that was added in the ER, right? It modifies that sugar to have this N6. So dedicated enzyme in the Golgi first recognizes the lysosomal hydrolases because of a specific signal patch they have. So it finds that signal patch, modifies the Golgi by adding on the N6P. Then that N6P, as it gets to the trans-Golgi network, there's a receptor that's like, aha, not so fast. It grabs it and helps ensure it gets trafficked to the lysosome. Good with me stuff? Okay. And here's an image of that, right? So here's our Golgi. Here's our lysosomal hydrolase. It's not showing the binding of the enzyme, but it's going to recognize the signal patch on that protein. It's going to modify the sugar so that it has N6P tag. that N6P tag gets recognized by the N6P receptor, which helps recruit the appropriate coat proteins and Rabs to make sure that this vesicle gets sorted to ultimately the lysosome, right? You're like, this is not math.
No, it's not,
Interesting if you want to learn about it. All right, so again here this is early endosome. So the part I want you to appreciate about this figure is that if this is binding to an early endosome, this is eventually going to take these proteins to a lysosome. Don't get too hung up on the endosome lysosome thing. I'm not testing you on it. I just want you to appreciate when you're like early endosome. What? I thought you said lysosome. This early endosome, the field thinks will develop into ultimately a lysosome. Yeah? Yeah.
M6P kind of just like a specialized signal receptor, specialized signal receptor protein type deal.
It's a specialized oligosaccharide, right, that is recognized by a protein.
Okay.
Yeah. All right. Yeah.
So if the receptor proteins aren't there, does it destroy the cell or do they just disintegrate away?
Oh, and what would happen if that receptor wasn't there? That's a good question. I don't think they get to leave the Golgi because it's the association with that receptor that triggers vesicle formation. Or they might start filling up in the Golgi and starting to break down the cell, trying to break down the Golgi. That's a good question. All right. The 3 types of endocytosis. 3 more slides. We're almost there. Probably not going to get to the end. It's okay. So endocytosis, inward flow, right? So there are 3 flavors: phagocytosis, pinocytosis, receptor-mediated endocytosis. So phagocytosis is when you're uptaking solid particles. Pinocytosis is uptaking fluid. Receptor-mediated means it's mediated by a receptor. So you have these receptors that when they bind to their ligand, that interaction triggers the formation of what's called a clathrin-coated pit. So you end up that ligand receptor interaction triggers clathrin's association at the plasma membrane, and it ends up forming a vesicle. So endocytosis, the molecule or those vesicles, they usually either go to the endosome, which is like the sorting station again, and sometimes components are trafficked back to the plasma membrane. So a lot of times what happens is whatever it bound to, it gets released in the endosome, like the ligand it got bound to is released in the endosome, but the receptor itself gets trafficked back to the membrane, for example. So all sorts of fun things can happen in the endosome. So it sorts the components that get back. And what coat protein is doing that job again? Clathrin, we got it. So here's just a cool actual picture of a coat protein forming in a vesicle. And then what's going to be right there? Diamond, you got it. Oh gosh, we have you're like, stop it, no! Dude, it's super easy though. It's just getting into the mitochondria, right? So you know this part. It's on. Really? Okay, well, we'll do on this on the side. We'll all just have to decide later.